Donc maintenant, on va pouvoir recevoir la deuxième récipiendaire du prix OSCAR 2022, donc Caroline Caetano Da Silva, qui est étudiante en thèse à l'Université de Paris, à l'unité INSERM Bioscar 1132, qui a débuté des études à l'Université de Sao Paulo avant de recevoir en 2019 donc une bourse Marie Curie, qui lui ont permis donc, de réaliser ses travaux de thèse sur l'ostéoporose idiopathique. Et elle va nous présenter aujourd'hui ses travaux portant sur la voie Wnt11.
Well, sorry, I was speaking English, but thank you very much for the opportunity to present the biggest part of my PhD project here, which was done at Bioscar in CERN 1132. And it's titled Wnt11, a new gene associated with early onset osteoporosis is required for osteoblastogenesis. So osteoporosis is a multifactorial disease characterized by bone fragility that will affect 1 in 3 women after menopause and 1 in 5 men above 50 years old. But studies performed in twins, they have shown there is a high heritability regarding the BMD levels. And bone fractures in young people is mostly caused by high-impact trauma, lack of vitamin D, or even secondary chronic diseases and medications. If any of these are the cause, it could be genetics. So which comes to a different disease, which is primary osteoporosis, which is a rare bone disease that it's characterized with the increased risk of fractures and typically affect young adults and children. So a study performed in the lab a few years ago in a cohort of 124 patients, young patients with low BMD, with osteoporosis. We checked in an NGS panel of 16 known genes for bone fragility, and almost 50% of the cohort had a negative results for these known genes, which made us think that's probably new genes related to primary osteoporosis that could cause this disease. So the aim of this project was to validate the function of genes in bone fragility and also investigate if Wnt11 is responsible for the bone phenotype. So Wnt11 is a gene that's already been described to play a role in the development of skeletal, kidney, heart, and lung, and also been described to regulate the osteoblast maturation. But so far, no disease have been reported in this gene. So our story with Wnt11 started when a young patient, 4 years old, arrived with severe osteoporosis, with low BMD and fractures. And we searched in the bone fragility panel, and no gene was found. So we performed a trio exome sequencing, and we found a de novo mutation that was a duplication that leads to a frame shift. So we started to do our functional analysis, and we find really interesting results. So we included the Wnt11 in our NGS panel, and we found two other patients. Both are female. The first one also with fractures and low BMD, and her mother also carried the same missense variant and presents osteoporosis as well. And the third patient with not so low BMD, but very unusual fractures. So we performed our functional studies using U2S cells with the osteosarcoma osteoblast cell-like cells, and we generate, using CRISPR-Cas9, a mutation of 32 base pair deletion, which leads to a frame shift in the same region as the patient one present his mutation. And we confirm the mutation in heterozygosis by sequencing and also mRNA expression and by protein expression. Ah, voilà. No. Ah, voilà. Ah, I see. Okay, protein expression. And we checked the proliferation, characterizing the cells. We saw that there was a decrease in the proliferation, almost 30% decrease. And then we performed mineralization assay, differentiation mineralization, and we see that the control cells are much more able to form mineralized nodules than mutated cell lines, which almost 80% decrease in osteoblast differentiation. To confirm that this mutation was, the phenotype of the cells was done by the mutation, we check by proliferation We check if when we treat with Wnt11 recombinant protein, we could see a rescue. And we saw in the proliferation. And when we treat the control cells with Wnt11, no difference, no significant difference is seen. But then when we treat we performed also the mineralization. And when we treat the mutated cells, we don't see any significant difference. Probably because differentiation, mineralization is a much more complex process, and something else could be happening before that could explain this. To try to understand, we check the osteoblast differentiation markers in our mutant cell lines, and you see a decrease for the most of them. And then Wnt11 is a known gene to be an important activator of the Wnt non-canonical pathway. And we check the canonical and non-canonical pathway genes by RT-qPCR. And we saw a decrease in the both pathways. We also treated with Wnt11 recombinant protein, and we saw a rescue, an important rescue for the non-canonical genes, but not for the canonical one, except for axin-2, which axin-2 is an important Wnt canonical pathway, but is known as a repressor of the canonical pathway. So to try to understand a bit better what's happening in the Wnt canonical pathway, we performed immunofluorescence in our cells to check beta-catenin, which you can see in green. And when we don't treat the cells, we don't see much difference in the localization of the beta-catenin, but sides of the shape of the cells, they are in the same localization. But when we treat with Wnt3A, which is activator of the canonical pathway, we see that beta-catenin goes to the nucleus, which means the Wnt canonical pathway is activated. But we don't see the same thing for the mutant cells. And when we treat with Wnt5A, nothing happens. When we treat with Wnt11, also nothing happens. But then when we treat it with Wnt3A and Wnt11, we see an activation for the control and the mutant cells. To confirm these results, we perform a TCF-Lab reported system assay, which is to measure the Wnt canonical pathway activity. And we see the same thing, that when we treat with Wnt3A, the control cells, they are activated. The Wnt canonical pathway is activating control cells, but not in mutant ones. Wnt11, we don't see anything, but Wnt3A and Wnt11 together, we see an activation of both control and mutant cells. Also, Wnt11 has been reported to promote osteoblast maturation and mineralization through responding 2. And we decide to check other respondins in our cells. So there is 4 respondins, and we check by RT-PCR in our mutant cell lines. And you see the responding 2 is decreased, a significant decrease. We also checked its targets, and only LRGR5 is this decrease in the mutant cells. So we treat LRGR5, we treat the mutant cells and check the LRGR5 expression. After the treatment with Wnt3A, it's nothing happens, but when we treat Wnt5A and Wnt11, remember that both are known as Wnt non-canonical pathway activators, and we see a rescue for both, for this when we treat with both. To confirm that we see the mutation, to confirm what we see when we treat with responding 2, we also perform the beta-catenin immunofluorescence, and we can see that when it's treated with responding 2, we see activation of the Wnt canonical pathway in control and also in the mutant cells, which we confirm by the luciferase activity assay. So which means that Wnt11 regulates the Wnt canonical pathway and controls responding 2. So to conclude, this is the first time that a variant in Wnt11 has been described to any disease, and also Wnt11 has an effect in proliferation, mineralization, differentiation, and it acts in both Wnt signaling pathways. And Wnt11 is probably upstream Wnt3A. That's why when we treat with Wnt3A, we don't see any difference in our mutant cells. So we believe that when Wnt11 is knocked out, we know that there is a decrease in the non-canonical genes. As well, there is a decrease in the responding complex and a decrease for the canonical pathway genes, which also there is an impairment of the beta-catenin to go to the nucleus. And so non-canonical pathway controls the responding complex and also controls the Wnt canonical pathway. And this study has been published in the Human Molecular Genetics. And of course, this project I wasn't doing alone. There was many collaborators and many people that helped. And I would like to thank my supervisors and all the colleagues. And thank you very much for your attention.
Moi, j'en ai une petite. Merci pour la très belle présentation. Je me posais une petite question sur la régulation de l'axine 2, donc, que vous avez montré surexprimée par le traitement avec Wnt 11. Mais après, vous ne nous avez pas redit, quand il y avait le co-traitement Wnt 3, Wnt 11, est-ce qu'au final, elle a été dérégulée et que ça permettait d'avoir une signalisation normale ou pas?
On n'a pas vu. C'est l'axine 2, elle a if it comes back, so, je peux répondre en anglais, c'est plus facile pour moi. We don't know if the axine 2 is rescue after the treatment. We didn't check, but it's interesting that axine 2 is a repressor of the canonical pathway. That's why we think when it's Wnt 11 is dysregulated, we don't see also could be through this axine 2 that we don't see a rescue in the canonical pathway. So we don't know exactly how the Wnt non-canonical controls the canonical, but we believe it could be through axine 2.
Okay, thank you.